Very good. Well, thank you, everybody. I need to give you a little background on how we got to where we are today, because I think it's important to get the foundation of how this technology was built, to understand why it's here and where it's going. So Dyadic International was an industrial enzyme company for over 20 years, competing with Genentech, Novozymes, and the largest companies on the planet, where we needed to develop a technology platform that could produce proteins at enormous scale at very low cost. So we came to this industry in a completely different way. We were producing proteins for industrial enzymes to make cellulases, to wash blue jeans, to make biofuels, to do all kinds of wonderful things for feeding and fueling the world. Of course, the slogan for bio.org is feed, fuel, and heal. So this technology has already successfully commercially fed and fueled the world, and now our final frontier is to heal it. And we believe, this is the expression stream. So what are we doing here? We're trying to express proteins. And we think we've created a cell through serendipity, through great science, that allows us to do that more efficiently, more effectively, and not only just in a large-scale production, but also in discovery and development, where we can take things in microtiter dishes and produce them at high levels that allow you to look at the protein, characterize it, determine the properties, in a system that you're going to scale it. Because we came from manufacturing down to discovery rather than discovering things and trying to figure out how we're going to make them. When we pre-screen the world in the production host, we know that we can streamline the process of scaling it up and getting better surety of yield and productivity and low cost. So I go to this slide just because I think what we're doing as an industry is we're doing injustice to society by using the same thing, doing the same thing over and over and over again, and expecting different results. And I want everyone to think outside the box, because in reality, we need to make things better. We need to speed the development up, and we need to reduce the cost so we can bring affordable health care to people. I'm going to talk about, in this case, an average number of CHO cell expression. We think from an industry standard is around 4 grams per liter. We know there are people that say they can do 10 grams. We know there's people that say they can do 1 gram. I think reliably, that's a good average number. It takes 14 days. But it's been 3 decades that we've pushed CHO cells to get to where they are. And so we've pretty much plateaued, although they're continuing to make improvements, they're slower than they used to be. Which leads to massive scale production. Now here you have an example of Samsung's $735 million plant. And again, I think the pharmaceutical industry was born on small molecules, and we have a small molecule mentality of tanks. And what you do in small molecules, you put chemical A in, you get chemical B, and you always get chemical C, and you want to make more, you just build more tanks. In biology, which we all know, it's about yield, it's about cells. So if a cell is 10 times more productive than another cell, guess what? We can use smaller tanks to get the same yield. So we don't need to build this huge infrastructure if we actually move away from inefficient cells and more productive cells. And I think if we think outside the box again, we can do that. And so the industry finally is waking up and saying, we need to look for an alternative to CHO. 45% of the people surveyed last year in biomanufacturing said that they agreed that we're too reliant on CHO. And by the way, that's 45% of the people who responded, and almost 40% didn't respond. It was more like 66% of the responders thinking we need to do something different. And we think that Dyadic, through serendipity, kind of like penicillin with their fungus, we've created a way to make proteins at very large scale, very inexpensively. But the good news is we can scale it down because we don't need to grow at the 500,000 liter scale that we've been growing at for 2 decades. We can grow in a 2,000 liter single use reactor and get 5 to 10 times the protein out at much lower cost. So we think already, and as top pharma is already considering and looking at this system and playing with it and funding research and development, 2 of the top 10 pharmaceutical companies in the world already funding research and development, and we've had great success. We'll get into some of that and on the things we've done, because confidentiality prevents me to tell you what they've done. One name so far is Mitsubishi Tanabe Pharma. There's allowed us to use their name. They have challenging proteins and they need help. They've come to us to try to help them overcome those challenges. So I want to talk about the science, because in the end it's all about cells and the potential of the cell, and what you can do with the cell and how you can manipulate the cell. This is a 25-year-old journey. We're not a new company. It's not a new cell. It's just a new cell to your industry. We've only been here for a year and a half, and I think you'll see the results we've had in the last year and a half are phenomenal. But it stands to reason that if you took a filamentous fungi, which is what the industrial biotech companies use to produce massive amounts of low-cost proteins, because they're secretors. That's what they do in nature. CHO cells were forced to do that. So you think you can take this higher, and you can take it farther, and you can take it faster. And we've proven that because we've actually produced over 100 grams per liter of protein with 80% purity, which would be 80 grams per liter of a single enzyme in the system in 6 days on glucose, very low-cost media, and we'll talk about that. But what happened with C1 that sets it apart from other filamentous fungi, besides the fact we spent 2 decades of creating molecular tools, how to program it, and do all the things we need to do, is we changed the morphology of this fungus. Because when you typically look at a filamentous fungi, you look at it and you think about, oh, it's filamentous, it's viscous, it's difficult to work with. Those are the problems of the past. It has a dirty background, it makes a lot of proteins in the background, not just the one you want to make. So if you think about a filamentous fungi and you took a hair of a woman's head and you stretched it out, you have 2 tips, and you're secreting protein from the ends, because that's what fungi do. Through serendipity and random mutagenesis, which even today we don't know how and why it happened, we created whiskers. We have hundreds of tips in the same mass. The protein went up 300-fold, and the viscosity went from 5,000 centipoise to less than 10. That was a major breakthrough. That's what turned the lights on. That's what made us decide to make it a recombinant host. And that's what we did. By the way, that mutation happened in 1996. So that's a long time ago, and we still have no idea how or why. But we don't, it's like Coca-Cola, they have a secret sauce. In our case, nobody knows why, so we can duplicate it. We don't need to know why, because we already have it. We can make more of it. The next thing that happened from a scientific perspective is in the industrial enzyme business, you keep trying to make more of something. Every year, just like CHO, you try to improve yields time and time again. We pushed again random mutagenesis. This is all before the sequencing of the human genome. We wiped out the secretome of all the things we wanted to make. Serendipitously, we said, oh, geez, we have instead of blue dark halos for an azo-CMCase assay for cellulases, we had a white one. And what are we gonna do with this thing? We almost threw it out. But it actually ended up, we found out a new promoter and the way to turn that on, and we can make 80% pure proteins instead of 80% of background proteins that are only 20% of what you want. So the energy was going to everything you wanted it to do and cleaned up the background. And you'll see that later as well. And then we've taken it through the US FDA in the feed and the food industry. So we've done pathogenicity studies, toxicology studies, mice trials, everything else, and got a GRAS approval in 2009 for food and feed. And of course, there's more work to be done for pharma, but there's nothing in there inherently that we're worried about. It's just a matter of like Nike, let's just go do it. And so we need the pharmaceutical industry to think differently, and let's just go do it together. And that's what we're starting to do with the partners. So we can grow it in any size you can imagine. Of course, we don't need to grow it at that sizes. And we can grow it in half the time to a third of the time of a CHO cell. So not only can we make more, we can do it in less time, and we can do it with much, much lower cost media. I don't know what happened here. So this is an example of what we're reporting today is 1.7 grams per liter per day. So if you compare that to CHO at 4 grams per liter, that's somewhere around 5 times higher yield. So that's where we are today. We can make a stable cell line from once you give us the gene fragment in 12 weeks. So we've taken the sequence, we made constructs. We normally make 4 different constructs with different promoters, different secretion signals. We have the native promoters, we have synthetic promoters, and we created all kinds of tools. Those genetic elements are getting better, quicker and faster and cheaper every quarter. With Moore's law and synthetic biology, this is just the beginning of time. So what we're already doing today is going to get a lot better, a lot quicker, and we're seeing those results. So from a standpoint of markers, we don't need to take a marker and keep it in there. We can take it out. If you have an orthotrophic marker, we can put it integrated in a genome specifically to a specific site, which we call hotspot, which we've identified. 1 gene, 1 copy in 1 site, high productivity. That's what I'm showing here. We can also do oxytrophic. We can do random mutagenesis, multivite copies, if that's what somebody would like to do and you want to ramp it up higher. But for now, we're focused on 1 gene, 1 hotspot, every time, plug and play, let's go do it. And we can work in microtiter dishes. And again, because of that unique morphology, that change, that random mutagenesis, we can fit in microtech dish. And you can see here, that gives us very, very good, if you discover something, you can make it quicker and faster. I'm going to show you a slide here of a fermentation, because people think, oh, filamentous fungi, what they look like. And I think that's how it grows. That's complete misconception to everybody in this room. You think it would be thick and viscous, but it's very fluid. So it's an amazing, that again is because of morphological change of viscosity. So it's a very simple process. It's a fed batch process. We start where we take 70% of the fermenter volume, working volume, and we feed glucose. And we use very simple media. It's amino acids and glucose. And we can get, in the case of this particular example, 1.7 grams per liter per day. In this case, it was a 30-liter fermenter. And you end up with a little more working volume. So if the whole volume is like a 1.3-liter fermenter, you get 1.3 liters. So an example, which we don't share, is we had 10.9 grams per liter of protein, an example I'm at now, which is a couple week old, recent data. But we ended up with 14 grams of protein from the fermenter, because we actually fed batch and you filled up even higher. So here's an example of an FC fusion protein that we've done. So this was a very challenging protein, because of course it's a new system. Pharmaceutical companies aren't coming to us saying, hey, just let's do the average thing. Where can you help us overcome a challenge? In this case, they spent 2 years trying to make this FC fusion, and they got 2 grams per liter and degraded 50% of it with the proteases in the CHO background. And in our case, we were able to make 8.1 grams per liter in 12 weeks. That was a phenomenal result. This shows you that the binding and the heavy and light chain together, they fold properly. It's a 1-to-1 ratio, a little more on the light chain, which is normal. And we get good results from the binding as well, you'll see later. Here's an example of another monoclonal antibody, in this particular case, where we made 8 grams per liter. And again, these aren't optimized processes. This is plug and play. Give us a gene, pop it in, 12 weeks later you have your strength, making grams per liter per day. One of the things that I did skip and I didn't want to go back to because I think it's important, you can use this, again, in a microtiter dish for your discovery and your development. So without any optimization at all, just in our normal way of making clones, we're getting examples of 1 mg per 4 ml microtiter dish, so a 24-well plate. Each one of those holes, you can get 1 mg of protein, which is plenty for discovery and development early on. And if we work on optimizing that, we can shrink that time, we think, down to somewhere around 3 to 4 weeks to get that kind of volume from gene to protein. And even higher levels if we actually start feeding those little wells with glucose, which we don't do now in the way we do it. So whether it's a microtiter dish, a little fermenter, or a large-scale production, you can make all the production you can ever want at each stage along the way. Which again, higher probability of getting to where you want to go faster with less risk. Here's another example of a 9.3 grams per liter antibody. We run them in 1 liter, then we go to, in this particular case, I think it was a 10-liter vessel. So these are just examples of MABs that we've done. There's other examples as well. Here's an example of the cost. I mean, if you look at the media cost, it's almost non-existent compared to CHO media. I mean, it's a defined media. We got rid of yeast extract. When we started off a year and a half ago, we were using yeast extract. And we said, well, we don't want to do that. Farmer would prefer not to have it. So we now figured out which amino acids we can use in the defined media. And we have a defined process with no yeast extract. And we actually got better results. And again, this is just getting going. We're just learning how to grow it for MABs. We've grown it for years for obviously making enzymes, different media, different conditions. So complete synthetic media. And then the binding, we produced CHO cell proteins that we got from the pharmaceutical protein partners, and we put them in C1, and they're virtually identical. And this is on 2 different MABs showing you that the binding is virtually identical. So theoretically, one, even if you didn't want to use it today to make your final product, and you wanted to use it to discovery and development, you're getting virtually the same binding. So early on, you can take a look at things, screen things quicker, get quick looks at things, and move it over to CHO, kind of what you do with HEC to CHO. But we think, of course, in the end, you're going to want to keep it in this cell because it would be much more productive, much lower cost. And then this is an example. If you take 4 grams per liter, you need two 12,000 liter fermenters to make 800 grams of protein. In the case of C1, if we just get 10 grams per liter, which we're already there, you can use two 2,000 liter reactors and get 1.2 million. So 50% more protein out than you can out of 24,000 liters. And if we can get up to 15 grams per liter, you can end up with 900,000 grams. So you go from 24,000 liters to 2,000 liters in single-use reactors. I mean, that's monumental. So when you combine this cell with a single-use reactor and low-cost media with no viruses, because it's a fungi, we don't have to do viral inactivation, which saves you 2 steps, it's remarkable. The future of biomanufacturing is just going to be changed by taking the power of industrial biotech and bringing it here and using synthetic biology, which we'll talk about now, from glycoengineering the strain. So one of the things, like all cells, you have proteases. Proteases chew down proteins, not a good thing. So we've been knocking out proteases one by one. Now people have always thought, geez, you knock out proteases, you kill the cell, it's not viable, it's not healthy. In our case so far, we've knocked out 8 proteases, and actually what's happened is we've improved doubling time. We've 2-hour doubling time now in a filamentous fungi. So it's the same speed, maybe even a little quicker than yeast. So it's very fast. It's got protease minus proteins. And if we get a mad now, we find a clip, we can find a protease and knock it out very quickly. And we're not using CRISPR, by the way, to do any of this, although we could. And the reason we're not is from a commercial perspective, we in pharma companies don't want to pay royalties. So if we build it, the ship, and we make it a little slower, and we get to the end, the rainbow, we have a royalty-free system without worrying about whose IP is CRISPR, what kind of royalty rate is CRISPR. We're just keeping away from all that. So in the end, we can deliver lower cost products because of those things. And here's an example of the continued progress at pH 5, 6, 7, and 8 of the protease background. And with this isn't the wild type, that 4 knockout was already dramatically reduced. And the protease levels were through the roof originally on the strain. So this is major improvements just in the last year. So now let's talk about glycoengineering, because of course this is the Achilles heel. You know, we talked about the power of the cell, the productivity of the cell, the robustness of the cell, the versatility of the cell, the scalability, the low-cost media, all the wonderful things. The point here is this is nothing new. People have done glycoengineering of yeast. The advantage that C1 has, if you think about Pichia, and let's talk about glycophi, because of course they did this, and people went, well, what happened to glycophi? Why aren't they using Pichia? And the answer is yield. They did a great job of glycoengineering the cells, but in the end, the yield wasn't there. Who's going to change from CHO to a Pichia when there's no commercial advantage? Even if you're 10 or 20%, we're talking about 400 to 500% better, maybe 10 times better in cost structure. That's the reason why people will change. So the other thing is, if you look at Pichia, they're like man 35. They have O-glycosylation, they have hypermannoses. We start only at man 9. So we've gone from man 9 in one short step, we have 80% now man 3. Now one thing I do want to point out, to date, we've not had any O-glycosylation seen in C1 and any of the proteins produced, whether it's a therapeutic protein, whether it's a mAb, and we've looked. And we've not only the pharmaceutical partners looked, we actually sent it out and said, go find O-glycosylation protein to glycan labs, and so far we have not found any. So that was another problem with Pichia, and a problem with other cells is the O-glycans is a major issue. The good news is we don't have to engineer it out, it's not there in the first place. So we have less work to do to get there. So this is kind of the timeline that we're on right now of where we went. Again, in the biofuel world, biotech, cellulase enzymes, obviously we've been there for many, many years. In the biopharmaceuticals, we expect by the end of 2019 to have a fully glycoengineered C1 strain with human type glycosylation. But the thing about the glycoengineered strain is we're going to have a variety of cell lines with different glycopatterns. So whether you want to use it for a vaccine and you want to create immunogenicity, and we have data that shows we can actually make the flu vaccine not only cheaper but better, but you can get increased immunogenicity or reduce it, and everywhere in between. And people haven't had a cell line to look at, because if you put in CHO, you're getting CHO-like glycan. You don't know if some other structure might be better for one molecule or not. Here we'll have different cell lines with high productivity with all different forms. So leading our scientific effort are a lot of bright scientists, smart people. But we also have a board of directors with really experienced people, both for vaccines and antibodies. And so I'm just gonna point a few. Dr. Bose was a Purdue engineer, graduated from Purdue, worked at Pfizer 34 years in her MAD group, one of the leading scientists and leading people in business development and science. Barry Buckland spent 29 years at Merck, same kind of pedigree. He's in fact the chairman of the board of NIMBLE, which is the government industry CHO group. And in fact, what's the guy heading the CHO group doing on a microbial systems board? You might ask yourself a question. He's there because he sees where we're going. And so basically, it's all about lower CapEx, lower OpEx, lower media costs, higher purity, no viruses. So the aglycosylated proteins we can do today, the vaccines we can do today, the glyco-engineered MABs that you need we can do tomorrow. We're on that path, and I think we're on the path of success. And together we can change the game. And so we want you to think outside the box and actually adopt something new and novel that actually bring benefits to society. Thank you.
Thank you very much. Are there questions?
So, how long does it take if you want to do it in a microcytoplasm? It wasn't clear to me how long it takes to go from a clone to a protein if you're just playing around in a microcytoplate as opposed to, and does it take 15 weeks to do it in a microcytoplate?
Well, part of that 15 weeks is getting the gene sequenced by something like GeneArt. So that would shrink it down to 12. But to get it into a microplate, no, it takes like 2 weeks to do that. But then to be able to select, we're selecting for clones, for high producing clones.
So you don't have anything like transients where you can just do a quick expression in a few days and then test what's coming out. You have to go through the whole process.
Well,
let me rephrase the question. It sounds to me like it's once you've got your molecule, it's great to then produce it. But if you're interested in testing 100 or 1,000 different molecules, then this is not the system.
Well, actually, it's not the system from a time, but the system to produce more. So if you take an Excel and you get there in a week, right? You get 20 micrograms or 2 micrograms or 5 micrograms, and you got to keep doing it because you need more and more. But you're right, it's not a discovery tool today, but we're going to try to shrink that timeline, but it takes time to grow. But we've not focused on that. Today, we're going to try to actually work with pharma to make it quicker and faster so that you can, for example, if you grow it in 3 days instead of 7 days, you're going to get half the protein. So instead of getting a mix.
If you're just trying to see does it work or doesn't work, and that may not matter, you want to get a quick result.
Yeah, today it's not the thing to use for quick results fast, in days, weeks.
Okay, thank you. In the way of producing in the fermenter, what range of pH can you grow these cells?
Good question. Because it's very wide. So we can grow from pH 4 to pH 8. So it's much more flexible than a CHO cell would be. We can grow from 25C to 43C. So again, it's much more flexible in the terms of pH and temperature range. Is very broad. So I probably should have brought that out earlier. So I'm glad you asked that question. And that makes a difference depending on the protein.
Very persuasive, very nice talk. You made a few, I would say, slightly provocative statements, which I would like to make a comment on. I've been in the CHO industry for more than 35 years. I've predicted about 20 years ago we'll reach 1 gram per liter. It came true much faster than I thought. 10 years ago I said, yeah, we will get to 10 grams per liter. And today I would say we go to 30 grams per liter. So while you say this is plateauing, I don't believe it. The industry has shown how we go from a very low density culture in CHO to a much higher cell density culture. Now we are 10 grams per liter with about 30 million cells per milliliter, and we only use 3% of the bioreactor volume. And that's one of the opportunities, obviously, for the future. Microbial systems like yours, go to 30 or 40% biomass per reactor volume. We will go not to that level, but we will go to 10%. And with this, we reach easily other levels. Okay, that's one statement which I would just correct. Don't say the industry is sleeping or research is not doing anything more to get more. The other comment I have, are we really sure that upstream yield is the limitation in cost of goods. I have serious doubts because, even though you might have, luckily, hopefully, less purification steps, and I've seen that's very convincing, I still believe we need a lot of water to purify things. And we don't spend money anymore on cell culture medium. We spend much more money on downstream processing and just getting to the purity level we require today for pharmaceutical proteins. So I would say I'm very open-minded. I've been, if you want, thinking out of the box all my life. So I'm very eager to actually look at your product and your cell host system myself. But we should not underestimate also the conservatism in our industry. Things which work, they should be fast into clinic and so on. So the things will move forward, I'm sure. You need a pioneer. You need a pioneering product which brings you to that level where you convince not only us here in this room, but FDA and others, and the clinical use in the market. I'm very happy to help you if you want.
Right. Well, we use
and
all the
I
help
wish
we
you good
all
luck.
the help we can get. But we do have 2 top 10 pharmaceutical partners already funding research. And the only thing I can say about CHO cells is, of course, they work, they're effective. They're really not, we would never use a CHO cell in the industrial biotech space to make anything because we could never make it affordable. So you spent 30 years pushing it on what you're doing. We spent 1.5 years and we're already at 10 grams per liter in half the time with 1/10 the media process. So we have a lot of room to go as well. And we would offer and accept all the help we can get from anyone in the room and elsewhere to actually bring this to the market. Because in the end, it's about bringing affordable health care to people. And one of the things, that last thing I'd like to say is, pharma is worried about and really focusing on, let's find a new gene to make a new drug to prevent death or to, recess disease. And the point here is, 90% of the population isn't getting the treatments today because they're too expensive and people are dying. And we're all sitting here doing the same thing over and over again, expecting different results. When in fact, we can save more lives by just getting more medicine to more people now, as well as developing new things. And that's sort of the global thought. And so we're here to try to help happen, make it happen quicker, get it adopted sooner. And anybody that wants to help in that effort, we're thrilled to do it together. And thank you very much.
Thank you.